Él él es especialista en control biológico, como digo, pero en los últimos meses o tiempo ha estado dedicándose a una plaga que ya habréis oído hablar, que es un vector de, de una enfermedad muy importante que transmiten estos insectos, estas enfermedades han ocasionado daños muy importantes en la agricultura en los últimos años. Estoy hablando de Xylella fastidiosa y va a hablar del comportamiento de este insecto, de cómo se alimenta de las plantas, y también creo que va a hablar de Scaphoideus titanus, que es el vector de una enfermedad que también importa y y cada vez más en cultivo de viña. Así que ahora os dejo a Guillermo para que continúe su charla.
Gracias, Alberto. And hello, everyone. If I talk in the microphone like this distance, it's okay? I think yes. Okay. So, hello, everyone, and I'm thankful for this opportunity to present my last work that changed from my habit of research because, as Alberto told, I'm more from the biocontrol research. And thanks to the XIC and ICA for the welcoming of this last week and maybe for future collaboration with the group of Alberto Freires. So before to start discussing with leafhopper and Spittlebugs and the feeding behavior, I will just briefly uh, introduce myself for those who didn't know nothing about the control egg parasitoids or sting bugs, but I believe you are a few, very few in this team. So uh, I started my research experience in the city of Dijon, where I did my master thesis, mainly in grapevine on egg parasitoids of Lobesia botrana, and the study of how temperature could modificate the relationship between the parasitoids, the pest, and the grapevine. And as Alberto told you, my main first work was to working on this very cute couple of host parasitoid, very unique in the world because this sting bug, Bagrada hilaris, uh, has this very funny behavior to bury its eggs in the soil. And this parasitoid, Graham Ethereum, formerly Graham gonicopalens, has this unique behavior to dig in the soil to look for the eggs. So it is a very beautiful coevolution and very interesting study model. And then I moved in the southwest of France to work on a more applicative way in biocontrol. So I developed a 3 years biocontrol program using Trisulcus species against the invasive pest Haliomorphalis that you, I believe, know all, and especially in the culture of hazelnut in the southwest of France because it is one of the main culture. And then I abandoned or I opened my mind to new research field, and I switched from biocontrol to the study of the insect vector, phytophagous insect vectors. So I'm quite new in this research field, and the results I will discuss with you are very new things to me, uh, as well as these two insects that we will discuss, so that you know, at least for one of them, Philenus pomarius and Scaphoideus titanus. So we can go in this insights in their feeding behavior. So a brief uh, recording of remembering of their biological cycle and their behavior in the field. So Philenus pomarius had this life cycle of laying eggs late in autumn and during winter, and overwinter as egg. And when it hatches in the late winter and the early spring, the larvae spend most of their time in the herbacee structs. So it can be found most at the ground level. And since it becomes adult, it starts to move to the trees, and especially the trees of importance, of agricultural importance for us. I mean olive trees, but also vineyards, as it is in the USA. And this insect is, as you know, a vector of the bacteria Xylella fastidiosa that came from the US and South America and arrived, 15 years ago in Europe, and started to be a big problem in our agriculture, especially in Italy, where I work now, who has two of these main culture of importance, grapevine and olive, that can be the target of Xylella fastidiosa. So this bacteria is a semi-persistent pathogen that only develops in the xylem of the plants it colonizes, and is vehiculated by Philenus primarius when it moves from a tree to another tree, inoculating the bacteria to its plant host. And in Italy in particular, so we have very big injuries on the trees in the south of Italy, in particularly on olive culture. But as I said, it can also damages the culture of grapevine and other fruits culture like citrus. On the other hand, we have Scaphoideus titanus and a closely related species of Fastidiosa variegatus that I will talk later, that are a lot different from Philenus primarius as vector, and also regarding the biological cycle, because this species is not polyphagous like Philenus primarius, but nearly monophagous, and make all his life cycle on the grapevine, laying eggs in the wood of under the surface of the wood of the grapevine, and all the larvae and all the adults can do all the cycle on the grapevine. So there is no transfer from the herbaceous plants to the tree plants. And this is the vector of phytoplasma, which is the phytoplasma of Flavescence dorée, which is a persistent pathogen that will be acquired mainly by the larvae of Scaphoideus titanus, and that will circulate in all the hemolymph of the insect, so that the insect will be a vector for all its remaining life. And as for Philenus primarius, it will transmit pathogen from the plant to another by feeding, but this pathogen doesn't develop in the xylem, but in the phloem of the plants. And this insect is not a xylem feeder, but it is a phloem feeder or a xylem phloem feeder, as we'll see later. And since, like for Xylella fastidiosa, it does damages to the leaf and the fruits by the obstruction of the plant vessels, the phloem vessels, that limit the water and the nutrient progress in the tissues. So I mentioned Xylella variegatus because in Torino, and in general for working on the Flavescence dorée pathogen, Xylella variegatus is considered as a good model, insect model, for the transmission bioassay, because it is laboratory reliable. It is not monophagous like Scaphoideus titanus, so it can be studied on other plants than the grapevine, which is not so much convenient. And it can be studied on the faba bean plants, which is more easy to rear. And so it is very easy to perform a lot of experiments instead of Scaphoideus titanus, which is monovoltine, which must be field collected mostly at the egg stage and then reared in the laboratory, and is specialist of the grapevine, so it is easier to do all the experiment with the grapevine. So we have these two insects that we will discuss together today. each one with his set of host plants and his associated pathogen. And in the University of Torino, we have for each of these two insects many research direction to understand their behavior, to limit their impact on the in the culture. For Scaphoideus titanus, it is mainly working on the varietal resistance. Also try new insecticides and see how they can limit the propagation of pathogen or the dispersion of the insect in the field. Working also on more biocontrol-like method like entomopathogenic fungi, and of course studying their feeding behavior to understand better how the transmission of the pathogen is done and what mean we could use to limit this transmission. And on the other side, Philenus pomarius has also a great panel of research direction, like egg parasitoids, studying the insect virome, RNA interference, entomopathogenic fungi also, and of course the feeding behavior. So this item of feeding behavior will be our main topic today. And we have especially a lot of collaboration regarding Philenus pomarius, including the XIC and ICA, mainly on the Bexil project and on the SOS project. So we have all these collaborators. I don't have to present this slide, I believe, to you because you have already plenty of these cages of electrophotometry. But for those who never seen this at ICA, this is an electrophotometry system that allows us to understand the feeding behavior of the insects by observing their behavior as waveform on electronical waveforms on a screen. And we can start the discussion with Philenus pomarius now. Mainly discussing about the very recent project aiming at studying how biotic or abiotic factors can influence its feeding behavior. And it is a very new project that started last summer. And we mainly have three ideas to start with this topic, is to studying how the temperature can influence the feeding behavior of Philenus pomarius, how the host plant species can modulate also the response together with the temperature, and how the hydric stress of the plant can also modulate the behavior of Philenus pomarius. And we have some expectation, but are mainly hypotheses that we have to verify. And for instance, for the temperature, we can both think that it will increase the food intake because of a need of the insect that is stressed at high temperature, but also it could be reduced because of the fact that an insect is not using too much energy to move and to feed. So that was two directions of results that we could expect. Regarding the host plant temperature, we didn't have any expectation regarding the two plants we studied, grapevine and olive trees. That are the two main culture of interest for us in Italy regarding Philenus pomarius. And the last point won't be discussed today because it is not done yet, but it is in project for this summer to study how the water stress of the plant still on these two plants can influence the feeding behavior. So we worked with the varieties of Barbera for the grapevine and Arbequina for the olive trees. And we picked the insect from the field directly at the adult stage, and we kept them at 25 degrees, which is between the two temperatures that we wanted to test, 20 degrees and 30 degrees. And to try to avoid as much as possible stress of the plant, putting them at 20 degrees or at 30 degrees, we decided to acclimate them at first a low and a high temperature so that during the EPG session, we can limit the response of the plant to the temperature. And after that, we performed EPG recording of 6 hours, that it is usually done with Philinus primarius, both on olive and grapevine at both temperature, 20 and 30 degrees. So the first main result that we can discuss is the time repartition of the feeding behavior of Philinus primarius. And the first very interesting thing is that all this green part, which is the xylem ingestion, which is the main behavior of Philinus primarius, when we pass from 20 degrees to 30 degrees, it increases in proportion, okay? So it doesn't mean that the insects at this step feed more on xylem because you can increase the proportion, but it is still a proportion. So you can also decrease maybe the time passed or the number of events. So we have to check all the aspects of this modification. But it seems that some behaviors, like low frequency ingestion of xylem in yellow and the resting time in red, transfers into xylem ingestion and nearly disappear. And this effect is stronger on olive trees than on grapevine. And so it answers to our first interrogation of if the high temperature would increase the food intake or not. But at this step, we have to go further in the details. And so we can check all the behavior and the duration of the behavior. The total duration of this behavior include non-feeding and feeding phases. Regarding the non-feeding phases, we can see that they are reduced from 20 degrees to 30 degrees, both on olive and grapevine. And it is the case for both the pathway phase, where the insect is actively looking for xylem in the plant tissues, and the non-probing phase, where the insect does absolutely nothing. So it go in the same way than the proportion of the time spent on the xylem that increase. In here, we observe that in fact the non-feeding period are decreasing. And it is the same for the resting phase, the air phase, that is drastically reduced in olive trees, less on grapevine, but also this behavior is reduced so that the insect seems to spend more time in the xylem to feed. And this is what we observe, that effectively the time spent in the xylem increase both on olive trees and grapevine. And surprisingly, it is not the case of the low frequency xylem feeding that instead decreases. And this is what we saw on the proportion graphics, that all these behaviors seems to transfer on the active xylem ingestion and the rapid xylem ingestion, not on the low frequency that decreases instead. And we have several ideas of how it could be explained. The main one is that the thermic stress could increase the metabolism of the insect and the needs in water and in nutrients. And as you know, the xylem is poorly charged in nutrients, so the insect have to take much more xylem to sustain. And when we check for the number of feeding phases, in that case, it doesn't change. So it implies that the time spent feeding in the xylem increases, but not the number of events. So it can be traduced by less or the same number of events of feeding, but longer events. And as you know, a longer events of feeding is also an increase in the transmission frame of the pathogen, possibly. And so this is interesting regarding also the transmission of Xylella fastidiosa. And finally, when we look at the time to reach the first sustained xylem feeding, it is also interesting to observe that this time is reduced so that the insect go faster feeding. That could be also correlated with the increased need of feeding because of the stress of the temperature. Interestingly, I presented all these previous results mixing female and males, okay? But we studied both. And interestingly, some changes in the proportion or in the doing of feeding behavior are not the same regarding male and female, especially for the low frequency xylem ingestion and the resting time. That, as you can see, for the low frequency xylem ingestion doesn't change from 20 to 30 degrees, both on olive and grapevine in the females. Regarding the air behavior, it changes a lot on olive trees, but not on grapevine. And this is also this can be seen also on these proportion graphs. Instead, when we look at the males, all these behaviors, and both on olive trees and grapevine, drastically reduced. So it seems that the high temperature has a stronger effect on the males that will transfer more of their time from resting, non-probing, or low ingestion of xylem to this active ingestion of xylem instead of females. And that are quite the same regarding the temperature. So the main idea could be that females are more resources to face the thermic stress. This is one hypothesis, but we are open to your ideas if you have some. And so they can face easier the stress of the temperature. Okay. So at this step, to briefly conclude on the Philonus primarius, these first results, this first experiment that we conducted in Torino, showed that the increase of temperature reduced the non-probing phase and the low frequency probing phase, and increases instead the time spent at actively ingesting the xylem of the plants. And we highlight a strong effect of the host plant that increase the effect of the temperature. And all the effects we observed were stronger on the olive trees rather than the grapevine. And we also highlighted a difference between the two sexes that may be interesting regarding the transmission of Xylella fastidiosa. So in the future, as I told you in introduction, we plan to study how the water stress of the plant can influence also this feeding behavior, because it is not excluded that in our trials, the plant has anyway a kind of stress. So we have to elucidate this part of the problem. Also, we use fababine for the rearing of Philonus primarius, and it could be interesting to check what happened on this plant, which is drastically different in terms of physiology. And it could be also very interesting to study what happens with different phenology states of the insect or of the plant. Okay, I didn't lose anyone at this step. Okay, so we can switch to the second part of the discussion about Scaphoideus titanus and Escherichius variegatus. And the results that I will discuss with you this time are very new also for the University of Torino, and will participate to a project of description of the feeding behaviors in cicadelids in general, and more specifically on Scaphoideus titanus and Oschilius variegatus, because they remain the main issue in the North Italy rather than Phylenus plumarius. And the objective is also to develop a strong knowledge on these insect models. So the problem of the varietal resistance, of studying the varietal resistance to Flavescence dorée, is that this resistance can be due to two main aspects of the interaction between the plant and the insect. The first aspect is a resistance to the insect feeding behavior, so that the plant will be more, more hard to reach, will develop some defenses, and so they will be less applicable to the insect. And the second part of the resistance to the phytoplasma transmission is the resistance to the pathogen multiplication and propagation in plant tissues. Okay, and we have a lot of studies for many years now focused on the transmission itself and on the phytoplasma itself and how the plant react to the phytoplasma. But we have a big gap of knowledge about the feeding behavior of the vectors of cicadelids, and this is the reason why we have to understand better. So as for Phylenus plumarius, I will only focus on this part, and I won't talk about the pathogen itself. So we have these two insects, as I told you before. One is our model study, Oschilius variegatus, because it is very easy to manage in laboratory. And one is the target species, okay, on grapevine, but less digestible, and we can work with it only from May to September each year. So it limits a lot all the investigations. The main problem is that Oschilius variegatus is excellent in acquisition of the phytoplasma, and behaves the same in terms of transmission as Scaphoideus titanus. So it is a good model for the acquisition and transmission bioassays. But as I told you, we miss a lot of information about its feeding behavior. So we have to improve this. And the main objective of these studies is to better understand the model and better understand how this model can be transposed to the target species, Scaphoideus titanus, and also to highlight the behaviors associated with the phytoplasma transmission, and in general to progress in the understanding of the cicadelids' behavior as insect vectors. So the first objective is to validate Oschilius variegatus as an EPG model, okay, because as a vector for transmission trials it is, but it is not clear that we can use all the results regarding its feeding behavior to make some conclusion with Scaphoideus titanus. So we have to validate both the model and the target. The problem is that Scaphoideus titanus can feed both on grapevine and faba bean plant, but Oschilius cannot feed on grapevine and feeds instead of faba bean plants. So we have first to resolve this first point, because faba bean physiology is not the physiology of the grapevine. And also we have to elucidate the feeding behaviors of infected insects, because all the transmission trials are done with infected insects. So we have to see whether this infection changes something in the feeding behaviors. So the first step to us appears to be to compare Oschilius variegatus and Scaphoideus titanus on a common plant, the faba bean plant. And then, because the target is still the Scaphoideus titanus on grapevine, we have to compare how Scaphoideus titanus behave on faba bean plant and on grapevine. And finally, we have to compare how infected insects behave regarding non-infected insects to see if we find the same behaviors and if there is some behaviors associated with the transmission in a case and not in the other. So the first axis of result was to compare these two species, and if we check the proportion of insects doing one of these main behaviors, feeding in the xylem or feeding in the phloem, we see that the profile of these two insects is quite the same, with the same patterns and the same succession of patterns. Both insects feed always first in the xylem and then switch to phloem, and generally when it reaches the phloem ingestion, it remains forever in the phloem ingestion. But we can see that the proportion of insects doing a pathway or doing nothing is higher in Scaphoideus variegatus, or this blue part, than on Scaphoideus titanus. So Scaphoideus titanus seems to be better at reaching the xylem or the phloem than Scaphoideus variegatus. And when we check the duration of these main phases, the pathway, the xylem ingestion and the phloem ingestion, we see that effectively the pathway lasts less for Scaphoideus titanus, and the xylem ingestion is higher. However, because we are on faba bean plants at this step, it seems that Scaphoideus variegatus is more prone to feed in the phloem of faba bean respect to Scaphoideus titanus. So the plant model that goes with the insect model, the faba bean plant, seems to be not very appreciated by Scaphoideus titanus, and it feeds not as good as Scaphoideus variegatus. So this is one point to consider for the future discussions. Now, when we compare Scaphoideus titanus on faba bean plant and grapevine, we see that when we look at the general profile of its behavior patterns, the profile didn't change, but the proportion of insect doing pathway is much more higher on faba bean plant all along the recording than on grapevine, and it is and it can transmit the idea that the faba bean effectively is less appetent than the grapevine, which has sense because Scaphoideus is associated with the grapevine as host plant. And this is traduced pretty well when regarding the duration of these phases, because effectively on grapevine, the pathway duration is reduced compared to faba bean, and the xylem and phloem ingestion are higher on grapevine than faba bean. So we have here, we highlight here one of the big problem we have is that working on the plant model faba bean seems to be quite not difficult but tricky to go further with Scaphoideus titanus, even if it is the host plant of Scaphoideus variegatus, which is our insect model. And finally, regarding the phytoplasma infection effect, these are very, very preliminary because these results I obtained them 1 week ago. So it is very recent, but we already see that the proportion of insects that are able to reach the phloem tissues of the plant is drastically reduced when they are infected. And this seems logical since the phytoplasma of Flavescence dorée is not co-evolved with Scaphoideus variegatus. And we know already that infected insects have a fitness which is reduced, and this can impact their feeding behavior as well. So we expect more results, more precise results with the precise analysis of all the records that we've performed. on Euchelidus variegatus. And this year, we also plan to do the same experiment with Scaphoideus titanus to compare if the infection by the phytoplasma reduces as well the behavior of the target insect species. So now, to conclude on this part and in general, we must face two big problems. The big problems is that the transmission trials that have been always done require several insects and require several days to obtain a result, because the transmission rate of Euchelidus variegatus, but also of Scaphoideus titanus, they are quite the same, is pretty low. So we have to put several insects on the same plant to observe near 90% of transmission success. Okay, and we have to make the trials lasting like 1 week. The main difference between this and the EPG trials is the time scale. Okay, the EPG trials, EPG recording, are done on 8 hours with only 1 insect per plant. So it is very, very difficult, if not impossible, to directly correlate any feeding behavior that we could observe, like this one, which is presumably a salivation waveform associated with the transmission of the pathogen. And it is nearly impossible to directly correlate this behavior to the transmission success, because we don't know how many of these waveforms we have to wait before to obtain the transmission of the pathogen. And as I told, the transmission rate is anyway obtained on several days and with several insects. So it will require thousands of trials of plants, and with maybe tons of variations of EPG interruption after, I don't know, 1 waveform, 10 waveform, 100 waveforms, we don't know. So it is very difficult, and we need to observe more precisely the feeding behavior, and to describe more precisely the feeding behavior to understand better how it performs. And also, regarding the last results I showed you, it is strange that the transmission of the phytoplasma is associated with phloem ingestion and salivation, because it is the only way for the phytoplasma to pass from the salivary glands of the insect to the plant. But we see that infected Euchelidus variegatus feeds much less in the phloem of the plant than non-infected insects. And in our records, we are completely unable to find the salivation or the supposed salivation events that exist in Scaphoideus titanus. So the model insect of Euchelidus variegatus poses these two main problems: that it is strange that it feeds so low at so low rate in the phloem tissues. At the same time, we don't see salivation events, but all the transmission trials are successful. So we are clearly missing some information, and this is a reason why we developed this project, to describe better the behaviors. Because we can assume that it is only a statistical effect, that we put plenty of insects that transmit at a low rate the phytoplasma for a long period, and we obtain a transmission. But it doesn't say anything about the individual transmission and what behavior is associated with. So this is a work in progress. And to conclude this presentation, we can see that Euchelidus variegatus. Oh, okay. So, okay. Bon, I don't know. But anyway, so Euchelidus variegatus is behaves quite the same than Scaphoideus titanus, but we were able to see some differences. Anyway, no, not this one.
Seleccionar.
Yeah. This one. It's okay. Hop là.
Espera, dar la durita cuenta.
Sì. Poi questo lo chiudo. Hop, 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 hop. I was at the last. Okay. Okay, excuse me for the inconvenience. Okay, so yeah, both species, the model and the target behave quite the same. But the results obtained in fababine are not directly usable, transposable to the grapevine. So we have to continue to study both species in parallel. And more importantly, there is some missing behaviors that we observe in Scaphoideus that we are unable to observe in Oceolus variegatus that should be associated with the transmission of the pathogen, and we cannot find them. So we need more direct waveform correlation. I written correlation. Okay, so it's it was late yesterday. I mean, and so I'm here, and I was here with Nicola Bodino, my colleague, the last week, to share some knowledge from the XIC and ICA, and to share their expertise on the techniques that can allow us to correlate feeding behaviors with what they actually mean and where in the plant tissue they actually occur. And so we've tried some unfortunately unsuccessful trials, but hopeful regarding the colectomy, regarding the cryofixation of the stylet in the plant tissues. And we've worked mainly with the model insect Oceolus variegatus, but we aim in the future to transpose all these techniques and all this knowledge to the target species Scaphoideus titanus. So here we are. I think I am great in time. Alberto Ferreres. Okay, we are on time. So I like to thanks all of you for your attention. To thanks my lab and my team, Domenico Bosco Laboratory. And again, to thank you, XIC and ICA, for the warm welcoming of these 2 weeks and the opportunity to share all your knowledges and your wisdom about the EPG and all the things that we, I hope, bring back to Italy. Thank you very much.
And for questions, if you want. Thank you very much for your talk, and now we can have some minutes for discussion and some questions or comments that you might want to do to Guillaume. Sí.
You don't have to. It's okay.
Thanks. Thanks for the talk, Guillermo. I have not a question, but a comment, and it's about the experiment related with the feeding behavior of Philaenus spumarius, and it's a comment that we used to discuss about in the lab many times.
I guess.
And it's about the convenience to analyze the low frequency waveforms together, the silent ingestion waveform or not.
Mm-hmm.
Because actually we don't know the real biological meaning of the low frequency waveform. So at the beginning, this waveform was included in the silent ingestion phase. So I don't know if you have analyzed your data considering both waveforms together or not, and if the results can change according to the biological meaning. I mean.
Okay, I get it. We can,
because sometimes we discuss about this, and for example, with the Tamara and the Rocio experiment, we have some doubts about this.
I understand. So the point is that we are still quite, new in this research field regarding Philaenus spumarius. So it is clear that I based all my analysis on what is the more precise description of the waveforms regarding their feeding behaviors. So it includes to separate low frequency xylem ingestion and normal xylem ingestion. The biological meaning of this, or the biological relevance of this separation, I agree with you that I have no clues about that. It is not very clear. And I don't know if Xylella fastidiosa can transmit better with an insect that is more in the rapid xylem ingestion rather than an insect that will be more in the low frequency xylem ingestion. So regarding your question, mixing both behaviors won't change the results that we've obtained, in the meaning that it is true that all the parts on, okay, we have a part of the low frequency ingestion that transfer onto the normal xylem ingestion. Okay, it can be just the meaning that the insect is more stressed, and so it will increase the frequency of its feeding without any biological consequences. But it doesn't change the fact that all the non-probing or non-feeding behaviors also are reduced with the high temperature, producing an increase in the food intake and a more sustainable xylem ingestion, both on the two plant species. So I cannot answer you about the relevance, in fact, of separating the both behavior, but it can be done.
Yeah, just to comment here, if you put together, if you lump together the low frequency plus the XI, in grapevine it won't change, because as I see here in the graph,
yeah, nearly no.
it will be the same. Both together, LF plus XI, adding them together, you will see the same effect. In olive, there might be a change, because if you add the low frequency to the yellow to the green, you will get a similar value as if you add the yellow to the green. In olive, it might not be such a difference. But yeah, the point is here, low frequency, we think it's also xylem ingestion, but at a lower rate. I mean, perhaps it's in the intake frequency, or the ingestion frequency is reduced. So the volume of sap that is being ingested might be less in the low frequency. But yeah, this result is very interesting. My question here would be, why would the insect will spend more time in xylem ingestion when it's under high temperatures? Do you have a hypothesis?
Yeah, in fact, this is we had no precise expectancies before to start this experiment, as I told, because we could also have obtained the complete contrary of results, that insects with insects not moving, not feeding, not spending energy, because the high temperature is too much stressful. Okay, but we observed that it was not that; it was the increase of food intake. The main hypothesis I would propose is that because constant 30 degrees is very high anyway, and so the insect can have its metabolism that increased with all the physiological functions increasing, so that the need for water and for nutrients increase also. We cannot exclude, on the methodological point, we cannot exclude that the period of starvation that we performed before the experiment was more stressful for Philaenus plumarius at 30 degrees than 20 degrees. And this is something to take account as a possible limit of our methodology, because 1 hour at 30 degrees is much more stressful for the insect. That is more looking forward to feeding on the host plant. And this can also explain what the time to reach the xylem was reduced at 30 degrees and at 20 degrees. Instead, at 20 degrees, it is quite cold for Philaenus primarius. And so the metabolism can be reduced with a need of food intake and water intake reduced also. So this is the main hypothesis.
Okay, so you did the EPGs in different rooms or chambers, I don't know.
Yeah.
Where they were subjected during the whole process, not only
yeah.
during the EPG recording, but before during the starvation.
And I know that for this point, it is very difficult to not do anthropomorphism, but spending all the time with the Philaenus primarius at 30 degrees or 20 degrees myself, of course, there is a main idea that the stress and the need of water and nutrients is increased. That there can be other explanation that doesn't came to my mind now, but maybe it is also part of the plant response that we try to reduce at maximum by acclimation of the plants before the experiment, so that there is not a heat wave or a cold wave. And we try to isolate as much as possible the response of the insect in this experiment.
Yeah, this would be very interesting to do the same thing under water stress conditions, because when the temperature increases, the water potential will be more negative,
of course,
and the insect will be probably pumping at a higher rate. This is why you get the higher.
Exactly. Also, we have in mind maybe to directly observe the gene expression of some gene associated with stress response of the insect or of the plant, to see their expression and to directly correlate these behaviors to an effective or not increase of these gene expression that will be more accurate to translate the fact that it was actually stressful for the insect to be put at 30 degrees and feeding at this temperature for 6 hours.
Great. More questions? No more questions. Well, thank you very much for your talk.
Thank you all.